Can CRISPR reinvent man?
Man is reinventing man all the time. Most of our things that make us beyond our ancestors, wildly beyond our ancestors, are physics, like, let's say, jets and cell phones. A few of them are biological, like vaccines. They're surely reinventing humans, and certainly making them transhuman, and certainly gene editing and CRISPR can participate in those revolutions.
So 7 or 10 generations from now, where do you see man?
7 or 10 human generations from now is a truly long period of time. We're talking about 250 years. It's quite likely that we would be as unrecognizable to current humans as current humans would be unrecognizable to primitive tribes or our ancestors 250 years ago. Maybe even more so because things are accelerating now, and so 250 years today is a great deal more technology than going backwards. So we would have essentially anything that we can dream of in terms of biology and chemistry, we can probably achieve through molecular technologies like editing.
I understand you have a list of the top 10 genes that people would like to have or should like to have. What are those?
Well, the list is now a couple dozen genes that are examples of extreme values in the human population, or their resilience. You should be at risk for a certain disease, but you don't get it. Or they could, some of them are synthetic biology where they've been tested in animals, but they all have in common that they're not the sort of things that contribute to the middle of the bell curve for people. They're way out on the extremes, and they're things like extra strong bones, which can inspire treatments for osteoporosis. There's resistance to HIV or to norovirus flu, stomach flu, and there's resistance to coronary artery disease with extremely low cholesterol forms. The list goes on of things that are potentially either good drug targets, good gene therapies, but in a certain sense they all have in common their enhancements, like vaccines. There's things that we weren't necessarily born with in the ancient days.
And along the way to these progressions, one of the things that I think everyone knows is that we need to be watching for unintended consequences.
Right.
Of getting to those points.
Right. So all new technologies have unintended consequences that we need to be wary of. This requires conversations among a diverse set of stakeholders and would-be stakeholders to make sure we're not missing something, and constantly asking, well, how can we make sure the benefits outweigh the risks? And in the case of these enhancements, you have a slightly higher proof required because you're starting with somebody that's healthy. And so when you give a vaccine to a healthy person, you need to make sure it does even less harm than if you're saving someone who's going to be dying in a few weeks anyway. That's a lower bar for that second category.
Mm-hmm. Will we one day be preventing disease, or will we always be treating disease?
Well, I think some of the most amazing, humane, and cost-effective components of our medical system are already preventative. If you look at cancer, for example, we spend about 100 times more on cancer cures than on cancer prevention, but most of the really great additions of quality-adjusted life years is from prevention. So these are, for example, vaccines like vaccines against HPV, reducing environmental exposure, so to sunlight, to smoke, to chemicals in the water, and other preventions that have to do with genetic screening, so you can get certain parts of body parts removed that are at risk that are otherwise dispensable. And so all of those are huge impact on cancer, much bigger, I think, than the cures we have so far.
How far do you think gene editing will go what's the potential for gene editing to go in terms of treatment of cancer?
Cancer is tricky in that it evolves. It's Darwinian. It changes. So that the best is to prevent it from ever happening. So, second best is to treat it with a treatment that's also evolving. So the immune system. And then third would be an immune system that doesn't evolve. So something where you've used the lessons of the immune system, but you kind of lock them in. And that's what happens with immunotherapy and CAR T and stuff like that. There's a whole new possibility of neoantigens where each individual cancer has its own specific signature, and you can attack that. Again, not quite as good as something that evolves on its own, but maybe we could turn it into something like that. It has the very quirky side from a regulatory standpoint that it's essentially an N of one. Each person is different, and so you can't do clinical trials on that particular antigen because it may be the only one in the world. And so that might actually, ironically, make it easier to get approval. We'll see.
Are there types of diseases you think that won't be treatable by genetic editing and treatment that results from genetic editing?
Are the diseases that won't be treatable by editing? I mean, in principle, you would think that would be true. Like, for example, accidents, if you damage some organ. But that, ironically, even that might be treatable with organ transplants, which have been developed by editing. So, for example, companies like eGenesis are making pigs that are where all of their organs and all the standard organs are transplantable into humans. And those organs could be enhanced in various ways. What else might? So that's a kind of an exception that proves the rule is that there are very few things that where you can't have a genetic fix, even if it's not a genetic cause.
Well, let's go back to transplantation and eGenesis for a moment. Excuse me. You have used CRISPR, and this is my layperson's interpretation, but you've used CRISPR to disable viruses that in the past could have been transmitted from pigs to humans if pigs become viable to create tissues and other life-saving organs. Is that correct?
That's right.
And that's a pretty big advance.
Right. So with transplanting from animals like pigs to humans, the biggest challenge involved immune rejection. But another big problem that set back basically a $2 billion investment over the last 20 years was the fact that every organ of every pig releases viruses into a potentially immune suppressed recipient of that organ. And so we wanted to make sure that we did no harm by removing those endogenous viruses. They're built into the pig genome of every pig on the planet. And so we made the first pigs by genetic engineering, by CRISPR, that are missing that. There are no functional viruses of that type.
Can those pigs then reproduce other pigs? Will the next generation also lack those viruses?
Right. So we're engineering these pigs for the so that we are engineering the pigs so the production can be as easy as possible, basically like breeding regular pigs, but they are essentially inbred with respect to the traits we care about, the things that make them immune compatible with humans, the things that help with the blood compatibility and the lack of viruses. All of these they can breed in a completely normal way, and then we can harvest the organs and maybe in the future be able to preserve them.
So the offspring of the pigs that you're doing this work on will lack the viruses as well.
Right. So the offspring of the pigs that we've engineered will lack the viruses and the other immune components that are hazardous.
And your work is just to create these pigs, not to actually work on the transplantation issues itself. You're creating this test tube, as it were, for other scientists who are transplantation experts.
We have active collaborations with transplantation experts. For example, the Massachusetts General Hospital is already helping us with non-human primate preclinical trials.